So this will just be a very brief review for the test 4, which is tomorrow. I hope that nobody's going to start studying like right now because there's a lot of information. But for this review, it's just gonna take out like some of the most important things that you should be knowing by now, and just give you a quick review on what you should be expected to know by tomorrow's test. And it will be using the exactly same questions that I have in the test 4 review. So you just need to go through the test 4 review. If you know how to do every questions in the test 4 review, this review will just be a summary. Right? So first question is to list out the 7 properties of life, examples for each properties of life. You need to remember all the 7 properties of life. There's order, reproduction, growth and development, energy processing, response to the environment, regulation, and evolutionary adaptation. So if there is anything that does not meet this requirement, then they are not a living organism. For example, a table cannot reproduce to make another table. They don't grow and become bigger, and they don't process energy. That's why a table is not a living organism. Second question, list out the biological organization of life from biosphere to the atom and provide examples for each level. So it starts from biosphere and then to ecosystem, to community, population, organism, organ system, organ, tissue, cell, organelle, molecule, atom, right? Everything is made out of atom. A group of atom is a molecule. A group of molecule that is a specialized part of a cell is an organelle. And then if you have different organelles, it makes up a cell. A group of cells for the same specific function is a tissue. And then if you have a group of tissues performing a specific task, it's an organ. If you have different organs working together, it's an organ system. And then if you have a group of organ systems working together, you become an organism. So example will be a human or a dog. So if you have a group of humans working together, there will be a population. If you have a human and a dog, a puppy, a chinchilla, they live together, there will be a community. And then if you have different communities with the temperature or weather, water, things that are not living, right? Things that do not have a life, then you form an ecosystem. And then eventually, if you have different ecosystem working together, you form a biosphere, which is the planet Earth. Next, list out the 3 domains of life and the similarities and differences between the 3 major domains of life. You need to know that the 3 domains of life are archaea, bacteria, and eukarya. And archaea and bacteria are all prokaryotes, so basically they do not have a nucleus, while eukarya is eukaryotes, so they have a nucleus. And since they are all living organisms, right? Every living organism must either be an archaea, a bacteria, or a eukarya domain. So they must all made out of cells. The cell theories applies to everything that goes into archaea, bacteria, or eukarya. So since they are all living organisms, they are all made out of cells, they must have cell membrane. They all have cell membrane, and they have cytoplasm and genetic material. Okay. Next, the 4 major kingdoms of eukaryotes. You need to know that under eukarya family, the eukarya kingdom, there is protista, fungi, animalia, and plantae. Animalia is basically animal cell, and plantae is made out of plant cell. And we did not go detail into fungi, but you are definitely very familiar about protista, which is a unicellular eukaryotic cells that the classic example will be amoeba. So amoeba is a protista. Fungi, you're very familiar with it. Mushrooms, animalia, us, plantae. List out the 3 subatomic particles of an atom, their charge and location. So we know that atom is the basic unit of matter, and to make up an atom, you need to have 3 subatomic particles that makes an atom. You have proton, neutron, and electron. Proton and neutron are found in the nucleus, while electron is found in the shell surrounding the nucleus. Proton is positively charged, neutron is neutral, and electron is negatively charged. Next, calculate the atomic number, atomic mass, proton number, neutron number, and electron number. We know that atomic number is just the same as the proton number. It's like an index. So we organize every element that we know in the world by how many protons they have, and we give them the number. So if you are the first element, you must have 1 proton. If you are the 5th element, you have 5 protons. So atomic number is just a number that we give them, index number. Atomic mass is basically how much weight they have, right? Each atom has their own weight, and we define the atomic mass by proton and neutron, because only proton and neutron has mass in the nucleus, while electrons are weightless, massless. Their mass is negligible. It means that it's not very big. That affects the atomic mass, so we just ignore it. We don't calculate it. The proton number is the number of protons it has, the neutron is the number of neutrons it has, and the electron is the number of electrons it has, right? So for an atom to be an atom, it must be neutral. Since an atom is neutral, the proton must be equal to neutron number. The proton must be equal to electron, because each proton is positively charged and each electron is negatively charged. So if an atom has 10 protons, it must have 10 electrons to cancel the charge. Equal number of proton and electron make this atom neutral. Identify ions and isotopes, right? So you need to understand the differences between ions and isotopes. Ions is basically when an atom has an unequal proton and electron. Because proton is positively charged and electron is negatively charged. So if the number of protons and electrons are not equal, then they will have some net charge. Net charge is basically when you have more proton or more electron. So for example, if I have 10 proton, but I have 9 electron, so I have more proton than electron, that makes me positive 1 charge because I have more protons. So the net charge is 1 more positive. So that's how we find the net charge. And when you have a positive 1 net charge, we call it an ion. And it is a positively charged ion, so it's cation. So a positively charged ion is a cation. If you have more electron than proton, for example, I have 10 proton, but I have 11 electron, I will have a net charge of 1 negative. That makes me an anion. So anion is a negatively charged ion. And isotopes is basically when you have atom of different neutrons. So if you have different different number of neutrons, you're going to have different number of atomic mass. That makes your mass slightly different from other people, but other elements. But you're still the same element because you have the same number of proton. So number of proton is the same, but the number of neutron is going to be different. Understanding the octet rule and electron arrangement. So octet rule and electron arrangement is just a way of how we put electrons into their respective shell, right? We know that shell has different levels. The closest one is shell number 1, the second one is shell number 2. And it has the maximum number of electrons that we can put into each shell. So let's say if I have 11 electrons, so I can only put 2 electrons in the first shell, and then 8 electrons in the second shell. That makes it only 1 more electron that I can have, right? Because the total number of electrons is 11 electrons. So the third shell can only have 1 more electron. So this way of arranging the electrons in each respective shell is going to contribute to the electronegativity. Basically, how much I want electron, or how much does the element want, how much does the element or the atom wants electron to fill out their valence shell. Right? So if I only have 1 valence electron at the valence shell, I will need 7 electrons to fill out the shell. So this is going to make the atom very reluctant to take in electron, because it's going to have to take in 7 electrons. It will be much easier for this atom to just take this electron and give it to some other people. So now that makes the valence shell having 8 electrons. So you will be able to see how valence electron contributes to the electronegativity by just looking at whether it will give away more electron or whether the atom will take in valence electron. Not a big part of it, you just need to know how to arrange the electrons and find out the number of valence electron for this particular test. 9. Understand the differences between ionic bond and covalent bond, right? So the way how atoms going to achieve the stable octet rule, basically fill out the valence shell with the maximum number of electron, is by giving away electron or sharing electron. If they give electron away, if they give or donate or receive electron, that makes the atom having more electron or less electron than it previously had. That is going to create ions, right? So if I have a positively ion, cation, and a negatively charged ion, anion, they will have opposite charge that's going to attract them and they bonded together. This bonding relationship between a cation and an anion is what we call an ionic bond. Sometimes atom will want to share electron. They share electron by putting the electron in the middle. That is going to create a covalent bond. So a covalent bond is sharing of electron. But depends on the atom's electronegativity. Sometimes this electron will be shared unequally. So it's either going to be closer to this atom or closer to that atom. Depends on which atom is having a stronger electronegativity. But because of these differences in electronegativity, the electron is going to be closer. That makes the closer atom having a negative charge, right? So if electron is closer to this atom is going to become delta negative or partially negative. So now that gets it to polar covalent bond and non-polar covalent bond. Basically is when you have two atoms, they share electron, right? That's what a covalent bond means. But the electron is closer. This electron is closer to the atom that has a higher electronegativity. Electronegativity is basically that this atom really likes electron. He will take in the electron to closer to this particular atom. Well, this atom has a lower electronegativity, so it does not have the ability to keep the electron to itself. That makes this atom positively charged, right? Partially positive. And this atom partially negative. So when you have a partially charged atom, we call it polar. Polar is basically having opposite charges. Opposite charge. So if you have equal sharing of electron, right in the middle is non-polar covalent bond. So equal sharing. And polar covalent bond is unequal sharing. 11. Understanding how hydrogen bond is formed from differences in electronegativity and partial charge of a molecule. So a classic example that we will use is water, right? Water is made out of 2 hydrogen and 1 oxygen in the middle, and they form a water molecule by having covalent bonds, so a covalent bond. But oxygen is more electronegative than hydrogen, so oxygen is going to take in all the electrons to oxygen itself. And because of this, now oxygen is negatively charged, partially negatively charged, and hydrogen is partially positively charged. So if this water molecule come close to another water molecule that has this partially positively charged and partially negatively charged, this partially positive charge and partially negative charge is going to attract each other because of this opposite charge. So this attraction is what we call hydrogen bond. So hydrogen bond is not really a bond, it's more like an attraction. The stronger the attraction it is, the stronger the hydrogen bond. The stronger the hydrogen bond, the easier it is to stick them together. So now you need to be able to identify the covalent bond and hydrogen bond when you have a group of or many water molecules. So this is a water molecule, a water molecule, a water molecule, a water molecule, a water molecule. Inside the water molecule, you have this covalent bond, right? So this bond between the oxygen and hydrogen is a covalent bond. And if you wanted to be more specific, it is a polar covalent bond because of the electronegativity. So the bond or the attractions between water molecules is what we call the hydrogen bond due to the partially negative charge and partially positive charge of the water molecule. Next, explain how hydrogen bond contributes to the properties of water. So water has different properties: adhesion and cohesion, surface tension, high melting point and boiling point, low density of ice than water. So adhesion is basically water sticking to the other molecules, water with other molecules. And cohesion is basically water sticking to water, right? So if you have strong hydrogen bond, you have strong attractions between different molecules that makes you very sticky. Surface tension is a result of that because you are very sticky. So for example, the mantis, right? The mantis is a bug. It can walk on the surface of water because the water is very sticky to other water, and mantis is very light, so it will not break apart water and just fall into the water. It will be able to walk on the water. High melting point and boiling point. Because of this hydrogen bond, you need a lot of energy to break the bond in order to boil it or melt it. So it will absorb a lot of energy. So that's why you sweat. By sweating, water evaporates and absorbs the energy and cools you down. Low density of ice is because water and water, when they freeze, the hydrogen bonds is going to space out. So when it space out, you increase the volume, but you did not increase the mass. So mass over volume ratio is going to increase. Mass volume ratio is going to decrease. That decreases density. Okay. Identify solvents, solutes, and solution. So solvent is basically things that you dissolve into. Solute is the thing that you use to be dissolved. For example, sugar or salt. So when you dissolve sugar or salt in water, so this is H2O, I put in salt. So now I'm going to make a salt water. So this will become a solution, salt water or salty water, the same as sugar. So this will be a solution. Recognize the shape and structure of 4 biomolecules. So you learn 4 different types of biomolecules: carbohydrates, lipid, protein, and nucleic acid. Correct? So the way how we recognize carbohydrates is that carbohydrates is usually having a ring shapes, a 5-ring shape or 6-ring shapes, and lipid, the classic one that we'll be using is triglyceride that has a glycerol and fatty acids. Protein is made out of amino acids, and nucleic acid is made out of nucleotides. So each one of this is a nucleotide. The biomolecules that we learn are these 4, right? Carbs, lipid, protein, and nucleic acid. And we learn about the cho-cho-chom-chom, which means carbs are made up of carbon, hydrogen, and oxygen. Lipid is also made up of carbon, hydrogen, and oxygen. Protein is made up of carbon, hydrogen, oxygen, and nitrogen. And nucleic acid is made up of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Okay, so the examples of carbs are those that ends in O's. You also need to remember about the 4 polysaccharides, right? Such as starch, glycogen, chitin, and cellulose. Lipids, the examples that you learn are triglyceride, fat, oil, steroid, cholesterol. Proteins, enzyme, antibody. Nucleic acid, DNA, RNA. Carbs function is usually structure or energy storage. Lipids is mainly long-term energy storage. Protein, you learn about the 4 functions: structure, defense, repair, and functions, body functions. And nucleic acids, genetic information. Recognize the reactant and product from equation or scenario. So basically, when you have a reaction, you take in something and you make something, right? The thing that you take in order to go through some sort of reaction is what we call reactants, and the thing that you make after the reaction is what we call a product. For example, you learn two different types of reaction. One is dehydration synthesis and one is hydrolysis. So dehydration synthesis or hydrolysis are example of reactions. So reactants are things that we take in order to make a product. For dehydration synthesis, the thing that you are trying to make are polymer. So you take in monomer and you wanted to make polymer. Hydrolysis is a reaction where you take in a polymer and you break it down with the use of water in order to make monomer. So recognize that as I explained the differences between dehydration synthesis and hydrolysis. I explained it before. Dehydration synthesis, you wanted to make polymer, and hydrolysis, you're trying to break down polymer. So you break down polymer into its respective monomer. So let's say if I have an example like glucose added with another glucose, they are going to go through a linking process, and this process is going to make a maltose, and with this I remove a water from the glucose molecule, so I make a water. So now this glucose is your reactant, and your maltose and waters are your product. So this process is dehydration synthesis. So the way how we tell whether this is a dehydration synthesis is by looking at the location of water. If water is a product, then it's a dehydration synthesis. If water is a reactant, then it is hydrolysis. List out the different polysaccharides, lipids, and proteins and their respective function. I think I go through it in slide 16. But it's always very good to remember the four examples of polysaccharides: the starch, the cellulose, chitin, and glycogen. Cellulose is used to make your cell wall. Starch is the food sources in plants. Chitin is the cell wall of fungi and exoskeleton. And glycogen is the food storage of animal. Or energy storage. Sorry, not food storage. Energy storage. Energy storage for animal. Saturated and unsaturated fats. The way how we tell is you look at the hydrocarbon chain. So hydrocarbon is basically a long chain of molecule that is purely made of carbon and hydrogen. So if you found that there is a double bond, right? As long as there's one double bond or triple bond, you're going to have unsaturated fatty acid. And if you see that the whole hydrocarbon chain is just filled with hydrogen and there's no double bond here, then this will be a saturated fatty acid. Saturated basically means that the whole bond, the whole chain is filled with hydrogen. Saturated is totally filled. There's no more space for new hydrogen to come in. While unsaturated, you have this double bond. I can still give hydrogen into this double bond. So that's how you tell saturated and unsaturated. The monomer and functional group of different polymers. We know that the monomer of this is amino acid. The polypeptide is the polymer version of protein. The monomer is amino acid. And this is DNA, right? So it's nucleic acid. The polynucleotide is made up of nucleotide. So nucleotide is the monomer of nucleic acid, and this is starch. So starch is made up of glucose or monosaccharide as the monomer of sugar or carbohydrates. And fatty acids does not really have monomer, it does have building block. So let's say the polymer that we're looking at is not the polymer, the biomolecule that we're looking at is a triglyceride. the building block would be glycerol and fatty acids. The glycosidic bond and peptide bond, so both of them are covalent bonds, but glycosidic bond is the bond that is formed between glucose, while peptide bond is the covalent bond formed between amino acid. Both of them are formed through the process of dehydration, and they can be broken down using the process hydrolysis. So dehydration forms glycosidic bond and peptide bond, while hydrolysis can attack it and break it apart. Enzyme, substrate, product, and active site. Enzyme is a type of protein, so it has 3D shape, and enzyme speeds up biological function, and that's why it is what we call a catalyst. It speeds out the reaction without actually being used. So the location with which the enzyme can put the substrates into is what we call the active site. The thing that the enzyme is going to bind towards to is what we call the substrates. Another word that you can say is the reactant. The things that you're going to stuff into the enzyme so that they can bind or do whatever the enzyme needs to do. Enzyme makes this reaction happen by lowering the activation energy. It makes it easier for the reaction to happen. And substrates. And when the reactions occur, you form a product. And this will leave the enzyme, and enzyme can be reused again. So activation energy, remember, is the energy that you need in order to change reactant into product. So let's say without the enzyme, right, the activation energy will be very, very high, and it will be very difficult for you to push the reactants to make product. But with the use of enzyme, the activation energy is going to be lower. That makes it easier for you to push the reactant to make products. And denaturation is a process where the protein or the enzyme is not functioning anymore. It will be affected by pH, temperature, and pressure, right? Because enzyme works in specific pH, temperature, and pressure. Without that, the 3D shape of the active site will be broken. You would no longer have that 3D shapes of the enzyme, that you will not be able to bind it with the substrates in order to proceed the reaction. So that's how these factors can affect and denature the enzyme. Similarities and differences between plant cell and animal cells. You should be very familiar. Animal cells does not have cell wall. Plant cell has cell wall, chloroplast, a large central vacuole, and animal cell has centrioles for cell divisions, and it does not have a cell wall. You need to be able to tell the differences between animal cell and plant cell and be able to label the organelles as well. And animal cells does not have fixed shape, and animal cell, you also need to know the similarities, right? They all are eukaryotic cells, so they all have membrane-bound organelles such as endoplasmic reticulum, ribosome, mitochondria, and nucleus. They are all cells, so they also have plasma membrane and cytoplasm. Where's the cytoplasm? It's not labeled. So the liquid between membrane and organelle is cytoplasm. You also need to know all the functions for each one of them. Okay. Eukaryotic cell and prokaryotic cell. Pro means before, eu means new. So karyote is nucleus. So eukaryotic cells have nucleus and all the membrane enclosed organelles, such as this is Golgi and then mitochondria. While bacterial cells or prokaryotic cells such as bacteria and archaea, they don't have nucleus and they only have membrane, cytoplasm, and genetic material. Both eukaryotic cell and prokaryotic cells also have ribosomes to make protein. So need to know the functions of them, but the key idea is that ribosomes makes protein, and endoplasmic reticulum, the rough one has a ribosome, so it will be able to transport the protein, and the smooth one will be used to detoxify the cell. Mitochondria is the powerhouse. Nucleus stores the genetic information. Plasma membrane protects the cell. Golgi apparatus package the protein. And centrioles is used in cell division. In plant cell, you have the cell wall to maintain its structure and shape so that it does not explode when this put in the hypertonic solutions. Vacuole is used to store water or other nutrients. Chloroplast is used to do photosynthesis. Okay, I think this is repeated. Yep. 29. A plasma membrane is made out of a structure called phospholipid. A phospholipid has a hydrophilic head. Basically, it is the phosphate group. It likes water. Hydrophobic tail does not like water. So you see that this whole region is made out of hydrophobic tail, which means that they don't like water, so you will not be able to find water. Water will not be able to exist in the phospholipid bilayers region, this particular region. No water, but water or other polar molecules will be at the extracellular space or the lumen of cell, basically your cytoplasm. And extracellular space that are outside of the cell. So you also have the proteins that are embedded in the phospholipid bilayer so that stuff can go in or out of the membrane. So the way how we do it is the facilitated diffusion, right? Facilitated means using or helping. So using protein to transport things to go through the membrane, because we know that membrane has this hydrophobic region. So usually only small and non-polar substances can be transported through the membrane through the movement of molecules. We call it diffusion, right? So diffusion means molecules move from a higher concentration to a lower concentration of that particular molecule. So let's say I want to talk about diffusion of salt. That it means that it will be a diffusion or movement of salt from a higher concentration of salt to a lower concentration of salt. So let's say if it is water, right? Diffusion of water, then it will mean it will be the movement of water from a higher concentration of water to a lower concentration of water. So the diffusion of water is basically mean a movement of water molecule. Another term for that is osmosis. And remember, all these things do not use energy because they all move from a higher concentration to a lower concentration, so they don't use energy. While active transport use energy in the form of ATP produced by the organelle called mitochondria, and it moves molecules from a lower concentration to a higher concentration. So let's say if it is an active transport of sugar. If it's an active transport of sugar, then it means it has to be moving from a lower concentration of sugar to a higher concentration of sugar. Diffusion, osmosis, and equilibrium. You need to know about the idea of semi-permeable membrane. It means that only selected stuff can move across this membrane. Usually big things like this sugar are not able to move across the membrane, while small things like water can move across the membrane. And they will always try to move around in order to reach equilibrium. Basically, it has the same concentration at both sides, equal concentration. But that does not mean that that does not mean that molecules stop moving. Things still move around. It's just that the rate of forward moving is the same as the rate of backward moving. So they still move across the membrane. It's just that you no longer see the changes on the movement as they reach equilibrium. So we can see that this region has a high concentration of sugar, very, very concentrated. So water will start diffuse from a lower concentration of sugar to a higher concentration of sugar, or a high concentration of water to a low concentration of water. High concentration of water, we call it hypo, while low concentration of water, we call it hyper, until they reach equilibrium. When they reach equilibrium, both of these solutions will become isotonic, right? So the hypertonic, isotonic, hypertonic. So you can see that in this picture, water move in and out, water move in and out. No changes occur to the cell. So this solution is an isotonic solution, equal solutions. You can see in this picture, Waters only enter it. This is the net movement of water because they are now put in hypotonic solutions, very high concentration of water. So water will start gushing into the cell, and animal cell will bust because they don't have cell wall, while plant cell will become very turgid, full with water because it has cell walls protecting its shape. If you put cells into a hypertonic solution, basically it's full with salt or solutes, very high concentration of solutes, water is going to start moving from within the cell to the outside of the cells because there's just low water concentration at the outside, so the cells will shrink. So in this one, this will be an example of the experiment that we did in class, right? During the lab, we have this dialysis tube where we put sugar and stuff inside the tube, and we put them into this beaker. Small things, like for example this red thing, they will be able to diffuse out, right? We know that small things will be able to diffuse through the membrane, while big things such as sugar, or all the different types of biomolecules that we have learned, like protein, they will get stuck in there. So remember, if I put big things into the tube, it will get stuck inside the tube, but if I put small things in the tube, it will start moving in and out of the tube through the semi-permeable membrane. So list down and identify the building blocks of a nucleotide. Nucleotide is the building block for nucleic acid. You had learned different types of nucleic acid, the DNA and the RNA, right? Nucleotide is made out of 3 different parts. The phosphate part, the pentyl sugar, it means it has 5 carbon, pent means 5, pentyl sugar, and the base. Learn the 4 types of base, the adenine, thymine, cytosine, and guanine, A, T, C, and G. And you learn 2 types of nucleic acid, the RNA and the DNA. RNA stands for ribonucleic acid, DNA stands for deoxyribonucleic acid. RNA is single strand, usually, most of the time. DNA is always double helix, at least in the human case. RNA does not have thymine, it has uracil. DNA does not have uracil, it has thymine. They are interchangeable. And RNA in human is split out into 3 major types you need to learn. The mRNA, which stands for messenger RNA, it transports information from nucleus to the cytoplasm. The tRNA, which stands for transfer RNA, it transfers the amino acids to the ribosomes to start making polypeptide. And rRNA, which stands for ribosomal RNA, that makes the ribosome in order to make the protein. And I think we repeated this again. Differences and similarities between RNA and DNA. No order basis, DNA pacing. A always pairs with T, C always pairs with G, right? You can see that the shape pairs very perfectly, but T and G does not pair, A and C does not pair. This is when you are in the DNA double helix situations, right? DNA pairs with DNA. But when you are in the mRNA and DNA parts, DNA starts pairing with the mRNA, A still pairs with T, but mRNA does not have T, so A will have to pair with U, U pairs with A, T pairs with A, this is DNA, this is RNA, right? DNA has T, but RNA does not have T. So if DNA is G, it will pair with the RNA of C. When DNA is at G, RNA will become G. So this is how you pair DNA to mRNA. A pairs with T, G pairs with C, A pairs with U, T pairs with A, C pairs with G, G pairs with C. So transcription and translations, we need to make protein from DNA, but DNA is in the nucleus. So first you have to get the information from the nucleus to the cytoplasm where ribosome is at. So you have nucleus, DNA is inside the nucleus, it starts getting the gene through mRNA, through the process of transcription. So with the process of transcription, so I have to put it like this. This is in the nucleus. Transcription is a process of making an mRNA from the DNA. So when you have the mRNA is going to go out of the nucleus into the cytoplasm, meet with ribosomes, and initiate the process of translations, where ribosome is going to use this mRNA to make protein. right? By taking the amino acid and bond them together. Cells are different in our body despite having the same DNA code because we have this process called gene regulation, where different genes will get turned on or off, depends on what cells you are. That's how you get so many different types of cells, right? You are one person, all your cells have the same DNA, same genetic information, but because cells are specialized, they do different things. Your stomach cells is not going to do the things your skin cell does. It will be very disgusting if your skin cells start producing gastric acids like out of sudden. So that's the fantastic part about your body and different types of cells. It's because of gene regulation. It makes different protein based on what cells it is. And be able to transcribe and translate a DNA code into a chain of amino acids. So let's say if your DNA is GCC-TAT-AGC. So you need to start doing the process of transcription, basically making an mRNA. G will have to pair with C, C will pair with G, C will pair with G, T, A pairs with U instead of T in the mRNA, T pairs with A pairs with U, G pairs with C, C pairs with G. And we need to be able to read mRNA in triplets, 3 codes at the same time. So CGG will be read as CGG, arginine. So you will be, the first amino acid will be arginine. The second one, AUA, so AUA is ILE, and UCG is serine. With that, hopefully you have a good revision.